When sequencing a typical bacterium, we might start off with a complete circular chromosome. Let's say we use the most common sequencing technology, which in 2020 is still Illumina. As Dr. Tucker described in his sequencing lecture, we end up with potentially millions of approximately 150 base pair fragments called reads. When we do genome assembly, we combine these reads into longer sequences, but we usually don't manage to reassemble a complete genome in its entirety. We can normally only build sequences that are shorter, contiguous sections of the genome, and we call these contigs. Even though we might have large contigs, and they might, sometimes, cover the complete genome, at this point we may not know what their correct orientation or order is, or how far away from each other they are. To link contigs together, we can use other information, such as paired end reads, which Dr. Tucker described earlier, or an existing reference genome to help with this. These kinds of data match two or more contigs and fill in gaps without giving us the actual genome sequence in those gaps. When we use this information to order and orient contigs, the result is called a scaffold.